Polyvagal theory, a primer. This is chapter 4 from Porges and Dana, Clinical Applications of the Polyvagal Theory, the Emergence of Polyvagal-Informed Therapies. Abstract. This chapter provides an overview of the polyvagal theory, and the chapter is organized to facilitate an understanding of the constructs embedded in the theory that are relevant for clinical application. The chapter is organized with headings identifying important constructs within the theory, many of which are used by the authors of the chapters that follow. Overview. Although polyvagal theory is the focus of this volume, the contributing authors were requested not to restate the details of the theory within their chapters. Instead, this chapter provides an overview of the theory. Although this chapter is dense and scientific, we hope that by providing information on the theory in the beginning of the book, we would allow the authors to be less encumbered by the science and to write in a more personal voice that would cover how polyvagal theory influenced their work. To facilitate the generalizability of polyvagal theory to clinical application, the chapter is organized with headings identifying important constructs within the theory. Polyvagal theory describes an autonomic nervous system that is influenced by the central nervous system and responds to signals from both the environment and bodily organs. The theory emphasizes that the human autonomic nervous system has a predictable pattern of reactivity, which is dependent on neuroanatomical and neurophysiological changes that occurred during evolution. Specifically, the theory focuses on the phylogenetic changes in the neural regulation of bodily organs during the evolutionary transition from ancient extinct reptiles to the earliest mammals. The evolution of the vertebrate autonomic nervous system. As mammals evolved, their behaviors differentiated from their primitive reptilian ancestors. Unlike the solitary behaviors and lack of nurturance of their vertebrate ancestors, mammals expressed a broad range of social behaviors, including caring for offspring and cooperation. These behaviors supported the survival of mammals. However, in order for these behaviors to occur, the mammalian nervous system had to selectively down-regulate defensive reactions. This convergence was dependent on the co-evolution of modifications in the neural regulation of the autonomic nervous system and the sociality that defines mammalian behavior. To understand polyvagal theory, it's first necessary to understand three contingent points. First, the relationship between autonomic state and defensive behaviors. Second, the changes that occurred during vertebrate evolution in the neural regulation of the autonomic nervous system. And third, the physiological state, which enables bodily responses and feelings of safety, optimizes social behavior, and concurrently optimizes health, growth, and restoration. In most vertebrates, the two primary defense systems are fight or flight and immobilization. Fight or flight behaviors enable the organism to flee or defend when threatened. These behaviors require the rapid accessibility of resources to mobilize through the activation of the metabolically costly sympathetic nervous system. Immobilization is a more ancient defense system that's shared with virtually all vertebrates. In contrast to the metabolically costly mobilization strategy, immobilization is an adaptive attempt to reduce metabolic demands, example, reduced options for food and oxygen, and to appear inanimate, death feigning. Juxtaposed with the rapid activation of the sympathetic nervous system required to promote fight or flight behaviors, immobilization defense behaviors required a passive shutting down of autonomic function via the vagal pathway within the parasympathetic nervous system. Over time, the second vagal pathway evolved and had the capacity to down-regulate both forms of defense. This second vagal pathway is observed in mammals and not reptiles. In addition, the anatomical structures regulating this component of the vagus interacted in the brain stem with structures regulating the striated muscles of the face and head to provide an integrated social engagement system. This emergent social engagement system provided the mechanism for co-regulation of physiological state, as mammals conveyed cues of safety and danger via vocalizations, head gestures, and facial expressions to conspecifics. The social engagement system enabled mammals to co-opt some of the features of the vertebrate defense systems to promote social interactions such as play and intimacy. These changes in the autonomic nervous system provided mammals with neuromechanisms to promote the biobehavioral states necessary for caring for offspring, reproducing, and cooperative behavior. In contrast, the adverse behavioral psychological effects of trauma appear to target a disruption of the social engagement system, its management of defense reactions, and its contribution to co-regulation and cooperative behaviors, including intimacy and play. Origins of polyvagal theory, the vagal paradox. Polyvagal theory emerged from research studying heart rate patterns in human fetuses and newborns. In obstetrics and neonatology, the massive slowing of heart rate, known as bradycardia, is a clinical index of risk and assumed to be mediated by the vagus. During bradycardia, heart rate is so slow that it no longer provides sufficient oxygenated blood to the brain, and this type of vagal influence on the fetal and neonatal heart could potentially be lethal. However, with the same clinical populations, the different index of vagal functions was assumed to be a measure of resilience. This measure was beat-to-beat heart rate variability and was the focus of my research for several decades. Animal research demonstrated that both signals could be disrupted by severing the vagal pathways to the heart or via pharmacological blockade, atropine, interfering with the inhibitory action of the vagus on the sinoatrial node. These observations pose the paradox of how cardiac vagal tone could be both a positive indicator of health when monitored with heart rate variability and a negative indicator of health when it manifests as bradycardia. The resolution to the paradox came from understanding how the neural regulation of the autonomic nervous system changed during evolution, especially through the transition from primitive extinct reptiles to mammals. During this transition, mammals evolved a second vagal motor pathway. This uniquely mammalian pathway is myelinated and conveys a respiratory rhythm to the heart's pacemaker, resulting in a rhythmic oscillation in heart rate at the frequency of spontaneous breathing, known as respiratory sinus arrhythmia. Myelin is a fatty substance that surrounds the fiber. Myelin provides electrical insulation for the fiber, which enables the signal to be transmitted with greater specificity and speed. This branch of the vagus originates in an area of the brain known as the nucleus ambiguus and travels primarily to organs above the diaphragm. It interacts within the brain stem with structures regulating the striated muscles of the face and head. The other vagal motor pathway does not have a respiratory rhythm and is observed in all vertebrae. It's unmyelinated, travels primarily to organs below the diaphragm, and originates in an area of the brain stem known as the dorsal nucleus of the vagus. Phylogenetic shifts in vertebrae, autonomic nervous systems. By tracking the evolutionary changes in the vertebrate autonomic nervous system, I identified a phylogenetic pattern consisting of 3 evolutionary stages. During the first stage, vertebrates relied on an unmyelinated vagus with motor pathways originated in an area of the brainstem resembling the dorsal vagal complex. During the second stage, the excitatory spinal sympathetic nervous system developed, which complemented the downregulation functions of the ancient vagal pathway. During the third stage, defined by the emergence of mammals, an additional vagal pathway evolved, during which cells of origin of the vagus migrated from the dorsal nucleus of the vagus to the nucleus ambiguus. Many of the vagal motor fibers originating in the nucleus ambiguus became myelinated and integrated into the function of the brainstem regulation of a family of motor pathways. Examples: special visceral efferent pathways that control the striated muscles of the face and head. In mammals, the unmyelinated vagal pathways originating in the dorsal nucleus of the vagus primarily regulate the organs below the diaphragm. Though some of the unmyelinated vagal fibers terminate on the heart's pacemaker, polyvagal theory hypothesizes that these unmyelinated vagal fibers primarily remain dormant until life threat, and that they are probably potentiated during hypoxia and states in which the influence of the myelinated vagal input to the heart is depressed. This sequence is observable in human fetal heart rate, in which bradycardia is more likely to occur when the tonic influence of the myelinated vagal pathway manifests in respiratory sinus arrhythmia is low. This also may be the mechanism mediating trauma-elicited dissociation, defecation, and syncope, fainting. In ancient vertebrae, an unmyelinated vagal pathway emerging from the brainstem was a critical component of the neuroregulation of the entire viscera. This bidirectional system reduced metabolic output when resources were low, such as during times of reduced oxygen, and the nervous systems of primitive vertebrates did not need much oxygen to survive, and could lower heart rate and metabolic demands when oxygen levels dropped. Thus, this circuit provided a conservation system that in mammals was adapted as a primitive defense system, manifesting as death feigning and trauma-driven responses as syncope and dissociation. Since this defense could be lethal in oxygen-demanding mammals, fortunately it functions as the last option for survival. The phylogenetically older unmyelinated vagal motor pathways are shared with most vertebrates, and in mammals, when not recruited as a defense system, they function to support health, growth, and restoration via neural regulation of subdiaphragmatic organs. Example, the internal organs below the diaphragm. The newer myelinated ventral vagal motor pathways regulate the supradiaphragmatic organs, the heart and lungs, and are integrated in the brainstem with structures that regulate the striated muscles of the face and head via special visceral efferent pathways, resulting in a functional social engagement system. This newer vagal circuit slows heart rate and supports the states of calmness required for social interactions. The ventral vagal circuit, coupled with other autonomic circuits, supports social play. Example, ventral vagal coupled with sympathetic activation and safe intimacy. Ventral vagal coupled with dorsal vagal circuit. Thus, the mammalian vagus has properties that promote states that contain the range of responding of all components of the autonomic nervous system and functionally restrains the system from moving into states of defense.